Hello, everybody, and welcome to today's webinar presented by Genex Technologies and Neoproteomics, entitled Fast Photochemical Oxidation of Proteins, or FPOP, Hydroxyl Radical Protein Footprinting, presented by Professor Joshua Sharp of the University of Mississippi. I'm Scott Weinberger, and I am your Master of Ceremonies, or host today. Today's session, as I said moments ago, is jointly presented and brought to you by the Alliance in Protein Footprinting that's been established between Genex Technologies Incorporated and Neoproteomics. We have formed this alliance to facilitate the rapid exchange of scholarly content and discourse among researchers such as yourself working in pharmaceutical and biopharmaceutical development and structural biological studies. Neo is a world-recognized leader in footprinting software and services for biopharmaceutical and structural biological research, and Genex is the world's only provider of footprinting instruments, software, and accessories that enable high-order structure analysis in a compact, intuitively simple, and benchtop format. Today's session is one portion of a 5-part scholarly seminar program that commenced with introductory talks by Professor Mark Chance and Joshua Sharp, who reviewed various different aspects of protein footprinting, as well as a discussion on protein footprinting applications in structural biology as provided by Professor Michael Gross of the Washington University of St. Louis. Both of these webinars were presented previously during the months of September and October, and they are available on demand right now at our website at genxtech.com. Today's session on fast photochemical oxidation, as I said moments ago, is being presented by Professor Joshua Sharp and represents the third installment of this 5-part series. And it is the first seminar that is actually providing an in-depth understanding or explanation of higher-order structural studies by protein footprinting, with a specific emphasis on the key factors for the design and successful execution of FPOP studies. Later in this series on December 9th, Professor Sharp will present a detailed discussion of hydroxyl radical protein footprinting data processing and higher order structure analytics, based upon which is fundamentally driven towards understanding the ins and outs of data processing and basically allowing you to have an appreciation of how to convert from protein structure mass spectrometry results to implications in terms of protein higher order structure. And the final installment of this session will be presented by Professor Lisa Jones of the University of Maryland on February 10th of next year at 11 a.m. Pacific Standard Time, when she reviews and provides a detailed discussion of in-cell and in vivo FPOP. Now a few words about today's speaker, Professor Joshua Sharp. Professor Sharp is a world-renowned and internationally recognized expert in hydroxyl radical protein footprinting. He has published more than 25 peer-reviewed articles in the field of HRPF, methods development, fundamentals, and applications between 2002 and today, with over 45 publications overall. Professor Sharp published the first benchtop methods for hydroxyl radical protein footprinting, including the first method using UV photolysis of hydrogen peroxide for hydroxyl radical generation. With numerous invited lectures to both academia and private industry on the application of HRPF to biotherapeutics, Dr. Sharp has addressed problems associated with formulation, biosimilarity, aggregation, protein-protein, and protein-ligand interaction mapping. Professor Sharp's lab is also known for their work in glycosaminoglycan structure functional analysis, and his team commonly leverages their expertise in both areas to study protein carbohydrate complexes. Professor Sharp received his PhD in 2003 from a joint program between Oak Ridge National Laboratory and the University of Tennessee, under the mentorship of Dr. Robert Hedich. He performed his postdoctoral studies at the National Institutes of Environmental Health Science in the Laboratory of Structural Biology under the supervision of Dr. Kenneth Tomer. In 2007, he joined the University of Georgia's Complex Carbohydrate Research Center as a research faculty member. Most recently, Professor Sharp joined the faculty at the University of Mississippi School of Pharmacy, where he currently serves as an associate professor of pharmacology and research professor of the Research Institute of Pharmaceutical Sciences. He has received multiple awards for excellence in research, most recently winning the New Investigator Researcher Award from the University of Mississippi School of Pharmacy and being designated at the University of Mississippi nominee for the 2019 Blavatnik Award in Life Sciences. Professor Sharp serves on multiple national and international advisory and review panels as an expert on protein and carbohydrate biophysical and structural analysis by mass spectrometry. It is now my sincere pleasure to introduce Professor Joshua Sharp.
All right, excellent. Thank you very much, Scott. I appreciate the kind introduction. Let's go ahead and get started. All right, can everyone see my screen okay? Yes?
Yeah.
All right,
good.
Excellent. Thank you very much. So, I am here today to talk to you all about fast photochemical oxidation of proteins, or FPOP, and its use in hydroxyl radical protein footprinting. Fast photochemical oxidation of proteins is one of a suite of methods that can be used to perform HRPF. If you need an introduction to what hydroxyl radical protein footprinting or covalent labeling in general is, or what it could be used for, I highly recommend you take a look at the two previous lectures in the series that Scott mentioned earlier, where we really get into that introduction and kind of give that overview. Today, what we're going to focus on really is getting down into the experimental details. I'm going to talk to you about the theory behind FPOP, how it works, and try to give you some kind of understanding about the various parameters that can be adjusted and some of the common pitfalls and practices that are used to help you, if you're interested, bring FPOP technology into your lab and get it set up and running with hopefully a minimal amount of growing pains. Just to give a real brief overview and refresher for everyone of what we're talking about when we're talking about hydroxyl radical protein footprinting. In hydroxyl radical protein footprinting, the idea of the experiment is to take a protein, expose it to hydroxyl radicals freely diffusing through aqueous solution, and then allow the hydroxyl radicals to react with amino acid side chains in the protein. When the hydroxyl radicals react with the side chain in the protein, they form stable oxidation products that we can then measure these stable oxidation products by mass spectrometry, with the idea being that by measuring these stable oxidation products, we can learn something about the structure of the protein. So on a fundamental level, what we're measuring is we're measuring these oxidation products, and fortunately, the stable oxidation products of most amino acid side chains with hydroxyl radicals result in a net mass shift that is identical between most of these amino acids. That is the net addition of one oxygen atom, which you can clearly see in a mass spectrum. So this allows us to actually identify and quantify these oxidation products pretty simply using standard LC-MS systems that most of us are familiar with. And the reason why we can get structural information from measuring this oxidation of different amino acid side chains really is at the heart of hydroxyl radical protein footprinting especially, but covalent labeling in general. And what we're really studying and measuring when we look at hydroxyl radical protein footprinting is we're measuring apparent rates of oxidation. So we're not just measuring what is being oxidized and what is not, but we're measuring how much of it is oxidized or how quickly is it oxidized. And the reason why we measure this is because the rate of oxidation of any given amino acid or region of a protein appears to primarily be the function of two major factors. The first factor is the chemical nature of the oxidized amino acid residue. Some residues are just inherently more reactive than others. So for example, sulfur-containing amino acids are very reactive, aromatic amino acids are very active, while small aliphatic and polar side chains are much less reactive. In addition, differences in the amino acid sequence context can make a particular amino acid target more or less reactive. So this chemical nature of the oxidized residue is one of the two factors. But the other major factor is the average accessibility of that amino acid residue, that oxidation target, to the hydroxyl radical diffusing through solution over the time of the radical exposure. And so we usually will simplify this into a solvent accessible surface area. So we'll say that the rate of oxidation is directly proportional to the average solvent accessible surface area, because the hydroxyl radical is about the size of a water molecule and it's diffusing through aqueous solution. So it's very similar to the average solvent accessible surface area. Now, since these are the two factors that we have to keep in mind, that both the chemical nature of the oxidation target and the average accessibility, the way the hydroxyl radical protein footprinting is commonly used is to compare two or more conformations or structural states of the same polypeptide chain. If you're looking at two structural states of the same polypeptide chain, the chemical nature of a given amino acid is identical because it's the same polypeptide chain. What can change is the average accessibility of that target to the solvent. And so we can interpret increases in the amount of oxidation in structure in conformation B versus conformation A. We can correlate that directly to increases in solvent accessibility and vice versa. So just again, as way of a very general review, a very brief review, the typical hydroxyl radical protein footprinting workflow is you have your protein in two or more structural states. Here we have ligand-free versus ligand-bound, and we expose it to hydroxyl radicals that we make using some method. In this case, we'll talk mostly about FPOP. These hydroxyl radicals will diffuse to the surface of the protein and react with amino acid side chains, leaving sites of oxidation, marked here with stars. And regions that have differences in their solvent accessibility will show differential labeling, either less labeling if it's shielded from solvent or more labeling if it's become more exposed. Now, these labeling sites, these oxidation products, are stable, which means that once we've completed labeling, we can then use our favorite digestion protocols, our favorite sample workups, and we end up with a suite of oxidized peptides where the information on the protein topography or surface has been frozen in the chemical snapshot that remains on the peptides themselves. So in areas where there were differences in the folded structure, we now see differences in oxidation in the resulting peptides. We then take these peptides, we measure them by LCMS, we quantify the amount of oxidation for each given peptide, and then we convert that into peptide level changes where we can see regions that become protected in the addition of, upon the addition of ligand, and possibly some regions that become exposed upon the addition of ligand. And then we can take that information, combine it with structural modeling, and find regions of ligand interaction and potentially regions of allosteric conformational change. In today's lecture, we are going to focus on the experimental workflow used in FPOP. So we're going to focus on this shaded area here. We will talk later in our next seminar about how we actually handle the data and interpret the data. Today we are going to focus on how do we generate the data, specifically using FPOP. And I want to give you a brief history of FPOP because I think that's going to help you appreciate why we do what we do in FPOP. So let me just give you a real brief history of benchtop hydroxyl radical protein footprinting, where it came from and how we got to where we are now. It's actually it's a relatively short history of what came before FPOP. We published the first benchtop mass spec-based hydroxyl radical protein footprinting back in 2003 using Fenton chemistry to generate hydroxyl radicals. So we would perform hydroxyl radical protein footprinting just in a test tube using chemical methods to generate these radicals. And it worked well, but it was a fairly dirty process and somewhat difficult to control. So in the next year, in 2004, we published a cleaner benchtop protocol using UV light and hydrogen peroxide to generate radicals. And the chemistry here is very simple and very easy to control. When we wanted to make radicals, we turned on a UV lamp. The UV light would photolyze hydrogen peroxide to form 2 moles of hydroxyl radicals per mole of peroxide. And then when we wanted to stop labeling, we would turn the lamp off. So it was very clean, very simple to control, and it generated enough hydroxyl radicals to actually label your protein. The big downside to this method, however, you can see in this figure on the right. So we had to put the protein in peroxide and let it sit, and that by itself gave us background oxidation. As you can see here, when we don't turn the lamp on, we still see some oxidation if we just let the protein sit for 30 minutes. When we turn the lamp on, we get a lot more oxidation, but to get this oxidation, we need to irradiate the sample, usually on the order of minutes. So it was a slow modification process. The reason why it matters that it's a slow modification process was because if you let this go longer, you start to see a distribution of oxidation products that looks like this bottom panel here. You see we've got one group of oxidation products here, and then we've got a second group of oxidation products here, okay, that are much more heavily oxidized. So what's going on here? Well, you start to see these bimodal distributions of oxidation products because you have to think back to what you're actually doing to the protein. And if you think back to your biochemistry, this starts to make sense. When you are probing the protein in hydroxyl radical protein footprinting, you're oxidizing the protein. And we know that oxidation is one of the major triggers for proteins to undergo conformational changes. Oxidation-induced unfolding, perhaps oxidation-induced aggregation, depending on the protein. And so we're starting, if you label for long enough, you'll start to unfold your protein, and you'll start to label the unfolded protein. And when this happens, you're introducing structural artifacts into your data because you're now probing a conformation that isn't the one that you care about. And that's what you can see going on in this panel here. This represents the mostly native conformation of the target protein, whereas this represents us probing a oxidatively unfolded conformation. And this is all occurring because it takes us a long time to actually generate these radicals. In 2005, Zay and co-workers published the first use of UV laser-induced hydrogen peroxide photolysis. It basically worked very similar to what we did in 2004, except now they're using a UV laser to do photolysis within a test tube. So they would put a microfuge tube in the path of a pulse laser, and they would shoot it with 1 to 100 pulses of UV light, depending on how much radical they wanted to make, and they would measure the oxidation products. And very shortly thereafter, just a few months after, Hambly and Gross published the use of a UV laser, this time coupled with a capillary flow system. And it's this combination of a pulsed UV light source combined with a capillary flow system that really is the hallmark of FPOP. Okay, so I want you to keep that in mind. Pulsed UV light source combined with a capillary flow system. So why is this important? What benefit do we get from using a pulsed UV light source with a capillary flow system? Well, the purpose of the capillary flow system is to ensure that each volume of sample that we irradiate gets hit with one and only one pulse of UV light. This allows us to very strictly constrain the lifetime of labeling, how long we label. And this is important because of the idea of the time scale of protein motion. Okay, so as I showed you before, if we label slowly, then if we want to prevent getting these oxidation-induced labeling artifacts, we have to label for a short period of time. And when we label for a short period of time, we don't get very much label introduced. And that's necessary, right? If we label for a long enough period of time to get enough oxidation, we're going to start getting oxidation-induced unfolding and labeling these unfolded conformations. So the problem we're trying to solve is there a way that we can label a protein heavily? Because remember, the more heavily we label the protein, the more structural information we get. So is there a way we can label the protein heavily without labeling the oxidation-induced conformations? And the way that FPOP tries to address this problem is by labeling the protein very, very fast. The idea being that if we label the protein, and the protein undergoes an oxidation-induced conformational change, as long as we don't label the protein again after it's had time to have that conformational change, then we're fine. We've still only labeled the native conformation that we care about. And so the analogy I like to use for this is a person who's having their photograph taken. So you stand in front of the photographer tells you to smile, you smile, they take the photograph, and the flash pops on the camera, and when the flash pops, you blink. Okay? But the idea is that by the time you blink, the photographer's already taken the photo. And so it doesn't matter. It's the same thing here. In FPOP, when you label the protein is going to unfold. It is going to have these oxidation-induced conformational changes, almost always. But the idea is that by the time the protein has had these conformational changes, you've stopped labeling. And in order to reach that, we really want to complete the labeling before the protein can undergo this folding in small peptides region, this helix coil transition. So what we're really trying to do is we're trying to complete labeling on the long nano to short microsecond timescale. If we can get labeling done in under 1 to 10 microseconds, then we should be good for labeling only the native structure, even if we label it very, very heavily and the protein unfolds afterwards. So that's why FPOP was invented. That's what FPOP strives to do. And today I'm going to talk to you about the experimental parameters that are needed to make FPOP work. And to do that, I've divided this lecture series into 3 different sections, 3 things that you really need to keep in mind when you think about how is my FPOP experiment going to work? How should I design this experiment? Okay, so to make FPOP work properly, you need to think about 3 different things. The first thing you need to think about is, how do I make hydroxyl radicals, and how many radicals am I going to make? So to make hydroxyl radicals in FPOP, you need 2 different things. You need hydrogen peroxide in some concentration in aqueous solution, and you need photons as measured as fluents, or photon density per unit area of your light cross-section. Okay, so the more hydrogen peroxide you have, the more photon density you have, the more hydroxyl radicals you make. The next thing you need to think about is the lifetime of labeling. Okay, how fast am I labeling? Because remember, we want to stay in this long nanosecond to short microsecond timescale regime. So how do we measure the lifetime of labeling? The lifetime of labeling primarily depends upon 3 things. The time frame of radical generation. How long am I making radicals? The peak concentration of hydroxyl radicals. What's the highest concentration of radicals I make? And then the half-life of the hydroxyl radical. How long do the radicals that I made stay in solution before they're consumed? And then finally, you need to think about the extent of labeling of FPOP. And the extent of labeling, or the number of oxidation events you get per protein molecule on average, depends largely on 3 things. The concentration of the protein analyte itself, the total amount of hydroxyl radical that you generate during the process, and then what competing reactions you have in solution that are consuming hydroxyl radicals other than the reaction of the hydroxyl radical with your analyte protein. And we're going to go over each of these steps as we describe the FPOP process and talk about some of the features and the problems that people typically run into. First, let me try to orient you by giving you a tour of what a modern FPOP workbench looks like. And so this is a photograph of the workbench that actually is in my lab right now. And I've divided this workbench into 2 different categories. There are the things you absolutely need to do FPOP, which are highlighted in red. And then there are the accessories that make your life much easier and make your experiments much more reproducible, which I've shown in green. Briefly, the way that the FPOP works is you have your protein in solution in a syringe with some concentration of hydrogen peroxide, as well as some concentration of glutamine and some concentration of adenine, and we'll go over what each of these is for later. And this solution is pushed through a fused silica capillary using a syringe pump. This capillary goes through the beam path of a KRF excimer laser, which fires at 248 nanometers. The laser pulses. It's focused by a plano-convex lens down onto an area of the capillary, which is the illuminated area. This is the region where the actual oxidation occurs. And then once the oxidation occurs, it's pushed out of the capillary and into your quenching solution, which contains usually methionine, amide, and catalase. Again, I'll tell you why this is important later. Some other accessories, so that's what's absolutely required to do FPOP. Syringe and syringe pump, fused silica capillary, laser, focusing lens. okay? But you'll see that's not all that we have on our bench. We also have an aperture. We have a motorized product collector. We have a motorized stage that controls our lens position. And we have a UV dosimeter. And again, I'll talk about why all of these accessories are useful. But for right now, I just wanted to kind of give you an overview. This is what an FPOP setup, a modern FPOP setup looks like. So if you were to build one in your lab, this is the kind of thing that you're going to try to reproduce. So let's start with our first step. Okay, the first thing we have to worry about in an FPOP experiment, making hydroxyl radicals. So remember, to make hydroxyl radicals, we need two things, hydrogen peroxide and photon density. The hydrogen peroxide is controlled very simply. What concentration of hydrogen peroxide do you put in your syringe? The more hydrogen peroxide you put in your syringe, the more hydroxyl radicals you're going to make, but there's a couple of things you need to be careful of. Remember, when I first introduced you to benchtop HRPF, I told you hydrogen peroxide by itself can directly oxidize proteins. Okay, it will directly oxidize via 2-electron oxidation, mostly at cysteines and methionines, although you can get some at tryptophans. Okay, so the good news is usually this reaction is pretty slow. So you can usually mix your protein with hydrogen peroxide on the order of minutes and not see much background oxidation. But the higher the concentration of peroxide you use, the faster the reaction goes. So as you increase your hydrogen peroxide concentration, you have to beware that you're not getting too much direct peroxide oxidation. Okay. The second thing you have to be careful of is if you add in way too much hydrogen peroxide, you can actually absorb so much of your photons that you end up getting low transmittance through the capillary. So most of your photons, or a significant portion of your photons, are being absorbed by the peroxide. The reason why that's a problem is because if your concentration is so high that you have significantly more photons at the front of your capillary than you do at the back, then you'll end up generating a gradient of hydroxyl radicals. You'll have more hydroxyl radicals on the side of the capillary close to the laser than you do on the side of the capillary far away from the laser. And this gradient in hydroxyl radical concentration is going to give you a gradient in protein oxidation, which is going to make things more difficult to interpret. So you usually want to keep your total transmittance high, generally greater than 90% total transmittance, and you have to be careful of direct peroxide oxidation as you change hydrogen peroxide concentration. But that's only one way that you can get more radicals. The other thing you can do to get more radicals is increase your photon density. Now keep in mind, photon density is not directly related to laser pulse energy. And that's shown here in this photograph we've got here. So with my excimer laser, with most excimer lasers, you can tell the laser, okay, how much energy do I want to deposit per pulse? The problem is, if I double my energy per pulse, that does not mean that I double my fluence, or my photons per square millimeter. And the reason why is shown here. On the left, we have the beam cross-section formed by an 80 millijoule per pulse laser shot. And on the right-hand side, we have the cross-section formed by 140 millijoule per pulse laser shot. And you can see the laser beam dimensions are significantly different. Okay, we've increased the laser beam width by about 50% after increasing the energy per pulse by just under 50%. So while we get some increase in fluence by increasing laser pulse energy, most of that pulse energy goes into making the beam larger, not making the fluence higher. So you have to be careful when you're changing and adjusting your pulse energy in order to change fluence. You have to be careful and always remember that changing the pulse energy is going to change the size of your beam. The other thing you can do to change your photon density is change the focal plane of your lens relative to your capillary. So if you bring the capillary closer to the focal plane, you focus the beam down tighter, you're going to increase your fluence. If you bring the capillary farther from the focal plane of the lens, the fluence is going to be lower. Okay, and that's where this motorized stage and this aperture come in handy. With the motorized stage, you can move the lens and therefore adjust the relative position of the capillary to the focal plane of the lens, which will allow you to pretty easily and directly adjust your fluence and change the concentration of hydroxyl radicals you're making. without having to fool with the amount of hydrogen peroxide that you have. Okay. So this animation is going to kind of give you an idea of how the FPOP flow system is supposed to work. We have our UV laser, and our UV laser is a pulse light source. So it's going to fire, and then it's going to turn off. And this pulse lasts about 10 to 20 nanoseconds, depending on which laser you have. So the pulse is very, very fast. And what you'll see is that we're going to illuminate an area of our flow system, and then we're not going to illuminate it again. We're going to turn off, and then after that area is passed out of the path of the laser, we're going to illuminate again, illuminating another area. But we have this region here that's not illuminated. Why do we have this region that's not illuminated, called the exclusion volume? The reason is that your proteins in the capillary don't exactly move like this. This is turbulent flow, where all parts of the capillary are moving at the same speed. That's not how capillaries work. The capillary actually moves according to what's known as laminar flow, where the sample that's close to the walls of the capillary is going to move more slowly, and the sample in the center of the capillary is going to move more quickly. And so that means that the front of your fluid is actually going to have this curved shape. So if we didn't have this exclusion volume, then we would end up double illuminating the more slowly moving molecules near the edge of the capillary. And if that volume that you're double illuminating gets large enough, you start to probe these oxidatively induced conformational changes. You start to see artifacts in your data. So we always have these exclusion zones calculated into our FPOP experiment. Usually these exclusion zones are about 15 to 30% of the total volume. If you want to see a more detailed treatment of why these exclusion zones are what they are, there are a couple of good papers I can recommend. The Gao et al. Paper is an empirical approach to the question of exclusion volume. And the Koderman paper is more of a theoretical approach to the question of exclusion volume and laminar flow effects. All right, but keep in mind, we need to have this exclusion zone or this exclusion volume to make sure that we're not double illuminating any of our sample. So how do we determine what our exclusion volume should be? To determine what our exclusion volume should be, we measure the width of the illuminated area of our capillary, and we use that to determine the volume illuminated per shot. And the volume illuminated per shot, the best way to think about it is you're measuring the volume of a cylinder. The height of the cylinder is the width of our illuminated area here. Okay, so the width of capillary that we're illuminating. And then the radius of our cylinder is 1/2 the inner diameter of your capillary. So you have illuminated volume is height times pi times r squared. That gives you the volume that you're illuminating with each laser shot. Once you have that, then you can use this formula to determine what syringe pump flow rate and what laser repetition rate do you need in order to get the exclusion fraction that you want. Okay, so the formula is pretty simple. It's flow rate times your inclusion fraction, or 1 minus your exclusion fraction. So what fraction of sample do you want to be illuminated, equals your illuminated volume per shot. So what volume am I illuminating for each shot versus how many shots am I taking in a minute? Okay, so based on the exclusion fraction that you want and the volume illuminated per shot that you have, you can adjust either your laser repetition rate or your flow rate to get that exclusion volume that you want. And this is where the aperture, one of those accessories, those helpful accessories I told you about, this is where the aperture really comes into play. If you don't have this aperture, then anytime you change the focal plane of your lens or you change the energy of your pulse, you're going to change the width that's being illuminated. And so you have to recalculate every time you change any of the parameters about your laser. But if you use this aperture, you can silhouette the laser beam that's impinging upon the capillary. So as long as you use this aperture, you know what this irradiated area is going to be, and so you don't need to recalculate your volume illuminated per shot. You always know that the width that it's going to be here is the width of your aperture. And so that's why the aperture is really helpful. It allows you to adjust your laser fluence without having to recalculate the volume illuminated per shot each time, and without having to adjust your flow rate or your laser repetition rate to keep a constant exclusion fraction. Okay, so we've talked about how do we make radical, how do we make more, how do we make less, how do we calculate our exclusion volume. The next thing we need to think about is the lifetime of labeling. So the lifetime of labeling in FPOP depends, again, remember, on three things. The time frame of radical generation, how long are we making radical? The peak concentration of hydroxyl radicals, how much are we making? And then the half-life of the hydroxyl radical. So usually when we talk about lifetime of labeling, we usually define that as the amount of time it takes until the concentration of hydroxyl radical drops below some defined level. And a defined level that's been used classically in the literature is 10% of the analyte protein concentration. So if your analyte is present at 1 micromolar, then you would say that FPOP is essentially done once the concentration of hydroxyl radical is below 100 nanomolar. Okay? But this is somewhat of an arbitrary cutoff. You could set this lifetime to whatever you want as long as you're clear about it. With the idea being once the hydroxyl radical concentration is below 10% of your analyte concentration, the amount of oxidation on the protein that you're going to see is going to be pretty negligible. Okay, so time frame of radical generation. For FPOP, this is not a problem. Okay, for FPOP, the time frame of radical generation is the width of the laser pulse, which remember is about 10 to 20 nanoseconds. Super, super fast. So this is not something we need to worry about in FPOP. What we do need to worry about is peak concentration of hydroxyl radical and half-life of hydroxyl radical. So peak concentration of hydroxyl radical is what we just talked about. How much radical are you going to make? And this is adjustable somewhat. But once you have your peak concentration of hydroxyl radical, then you need to think about how long is this hydroxyl radical going to last in solution. And when we think about that, there are a few different pathways of hydroxyl radical decay we need to keep into account. And I want to talk about three of them, or three different categories of hydroxyl radical decay that are important to keep in mind. And the first is the one that actually causes people the most trouble, which is you have to remember hydroxyl radicals react very, very efficiently with other hydroxyl radicals to recombine and reform hydrogen peroxide. So remember, this is going to go, the rate of this reaction is going to go up as the square of the hydroxyl radical concentration. Okay, so that means that as we make more radical, we're going to increase the amount of hydroxyl radical that's consumed by recombination faster than we increase the amount of hydroxyl radical that's consumed by other pathways. Okay, so you don't have a nice direct proportional correlation between how much radical you make and how much radical attacks your analyte, because the amount that you lose to recombination goes up as the square of the concentration. The second pathway of hydroxyl radical consumption is the useful one, when the hydroxyl radical reacts with your analyte protein to give you an analyte radical. Okay, this is what we care about. This is what we want to happen. And then the third one, and one that is always important to keep in mind, and always important to have at the front of your mind when you're designing your FPOP experiment, is what I call everything else. The hydroxyl radical reacting with anything in your solution that's not your analyte and not other hydroxyl radical. These are all scavengers. Now remember, part of why we like the hydroxyl radical is because it is a very, very powerful oxidant. But since it's such a powerful oxidant, that means that it will react with just about anything you put into solution, especially just any organic molecule you put into solution. So whenever you're thinking about your experiments, you need to think about all of the things that you're adding into solution and how that can affect the lifetime of your hydroxyl radical. Now, using this information, people have done simulations of the lifetime of hydroxyl radicals because we want to ensure that we keep the lifetime of hydroxyl radicals to that long nanosecond to short microsecond regime that we need to prevent labeling of artifactual conformations. And so if you just make a burst of hydroxyl radicals with nothing else in solution, so you make a 1 millimolar concentration of hydroxyl radicals with nothing else in solution, and you measure their decay, these hydroxyl radicals will persist in solution on the order of milliseconds, okay? Much longer than we really want for true FPOP labeling. So to compensate for that, traditionally in FPOP, people add in scavengers. Okay, other small molecules whose main job is just to react with hydroxyl radicals to keep this lifetime limited. The most commonly used scavenger is glutamine, but you can actually use anything you want, but you have to keep in mind the concentration that you're using and the reactivity, the inherent reactivity of your scavenger and the hydroxyl radical. 20 millimolar glutamine is nice because it limits the half-life of the radical to right around 1 microsecond, which is the target we want. If you use 20 millimolar phenylalanine, which is much more reactive, you actually get down below 1/10 of a microsecond. But you end up diverting a lot of your hydroxyl radical to the scavenger pathway, which means there's less around to go to the analyte. And remember, the less oxidation you have of your analyte, the less information you have. And so you're really looking for that happy balance between not enough scavenging and too much scavenging. Now, keep in mind, this simulation was done without taking into account this pathway, the reaction of hydroxyl radical with analyte. And depending on your analyte, this reaction can actually be very, very, very significant. A lot of analytes scavenge hydroxyl radicals extremely well. And so depending on what analyte you're using and what concentration of analyte you're looking at, you may not need to use any glutamine or any scavenger at all to keep your lifetime of your hydroxyl radical to the high nanosecond to low microsecond regime. This 20 millimolar glutamine or equivalent is basically just a safety feature. If you use this concentration of glutamine, then even if you get no reaction with your analyte, you know the lifetime of your hydroxyl radical won't be longer than 1 microsecond. Okay, and the last thing we want to keep in mind is the extent of FPOP labeling. And by extent of FPOP labeling, we mean how many labeling sites do we get per protein on average. The extent of FPOP labeling depends on 3 things. The first thing is the concentration of protein, and this is actually a little counterintuitive for some people. So if you just think back to your standard chemistry rate law, right? Velocity of a reaction. The reaction of a hydroxyl radical with a protein is a second-order reaction. The velocity of that reaction should depend on the concentration of the radical and the concentration of the protein. The higher the concentration of the protein, the faster the reaction goes. Okay, and that's true. That is completely true. But what it doesn't take into account is we don't care about the velocity of the reaction. We care about how many reactions occur on each molecule of protein. And when you calculate it that way, how many reactions occur on each molecule of protein, actually the lower your protein concentration, the more oxidation you get per protein. And so the more information you get, the more labeling sites you get. So this is a figure out of a paper from the Konerman lab where they looked at the oxidation of a protein at 3 different concentrations, 11 micromolar, 22 micromolar, and 44 micromolar. And as you can see, as the concentration doubled, the amount of oxidation per molecule on average decreased significantly. Okay, so concentration of protein in general, less is more. But remember, if you get too crazy and you drop the concentration of your protein too low, your sample losses that you get during handling are going to end up making it very difficult for you to detect and quantify your protein. So as a matter of practicality, generally speaking, we keep our protein in the range of 1 micromolar concentration. We can go higher, we can go lower, but usually our median is around 1 to 5 micromolar protein concentration to get good results. The second factor is the total amount of hydroxyl radical generated. If you make more radical, you're going to have more reaction with your analyte, all other things being equal. And then the third thing are, what are the competing reactions that consume the hydroxyl radicals you make? And I really want to talk about the interplay between these two. So let's do a quick simulation. Let me walk you through a quick simulation here. So I have a scavenger. I have a mixture, an FPOP mixture, okay? And this mixture contains an analyte protein, a scavenger, and my hydrogen peroxide. And I perform FPOP. FPOP, and I generate a peak concentration of 1 millimolar of hydroxyl radical. and I measure the amount of my scavenger that's oxidized by this hydroxyl radical, and that's shown here with this black line. So it gets up to some particular concentration. Okay? Now, if I take this same solution under the same FPOP conditions and I add in a scavenger, in this case I add in 1 millimolar histidine, now the hydroxyl radical can, in addition to reacting with a scavenger, it can now instead react with the histidine. So it has a competing pathway. And so the amount of oxidation of my scavenger is now considerably lower. It's this blue dash line here. But what I can do is I can keep this histidine in solution, but I can turn up the amount of hydroxyl radical I make, either by increasing my peroxide concentration or increasing my laser fluence. I can make more hydroxyl radical. And so if I take this same solution with the histidine, and instead of making 1 millimolar hydroxyl radical, I make 1.82 millimolar hydroxyl radical, now I can get back to my original level of oxidation of the scavenger. Okay, who cares, right? Sure, who cares though? Well, the reason why this is interesting is because if I take these same conditions and I look at the modification of my analyte, my protein analyte, so under conditions of no histidine, 1 millimolar hydroxyl radical, I get protein oxidation shown here in black. If I add in the histidine, the amount of oxidation of my protein drops, just like the scavenger did. And then if I turn the amount of hydroxyl radical up to 1.82 millimolar, where I get the same amount of oxidation in my scavenger, now if I look at my protein, I also get the same amount of oxidation in my protein as I did without the scavenger and with the lower amount of hydroxyl radical. So what that means is that I can monitor oxidation of a scavenger and use that as a readout of the amount of oxidation I have of my protein analyte. Not only can I use this as a readout, but if I see less oxidation of my scavenger, and therefore can infer less oxidation of my protein analyte, I can then compensate for that reduced oxidation by increasing the total amount of hydroxyl radical generated. And if I can compensate for the oxidation in the scavenger, I can infer that I have compensated for the oxidation in my protein analyte. And this is really the foundation behind one of the most important factors in FPOP and one of the areas where new people to the field most commonly make mistakes, and that is in the question of hydroxyl radical dosimetry. How do I measure not only how many radicals I'm making, but more importantly, how many radicals am I delivering to my protein analyte? Now, hydroxyl radical dosimetry is something that lots of different groups have worked on, and there are lots of different ways to do it. Mark Chance's group came up with a way for doing it when they're doing hydroxyl radical footprinting using synchrotron radiolysis, but unfortunately, this method does not work yet for FPOP. The Gross Lab published an isotope dilution GCMS method for dosimetry in FPOP. A couple of labs, including my lab, the Gross Lab, have published methods using internal reporter peptides for doing dosimetry. But the way I really want to talk to you about is the way that that we think is the easiest, the most informative, the fastest method for doing dosimetry. And that is doing dosimetry by UV absorbance, most typically using adenine. And we've published several papers on this. The way it works is really quite simple. So you have adenine in solution as one of the scavengers in your FPOP system. And adenine by itself has a very nice UV absorbance peak here around 260, 265 nanometers. And if you add in hydrogen peroxide, the absorbance stays the same. And if you shoot it with a laser, the absorbance stays the same. But if you add in hydrogen peroxide and shoot it with a laser, so if you oxidize it with hydroxyl radicals, this absorbance peak drops precipitously. And you lose most of your absorbance peak at 265 nanometers because the oxidation product of adenine does not absorb as strongly there. So what that means is that we can measure the oxidation of one of our scavengers using very simple photometric methods. And since, as I just showed you, since we can measure the oxidation of a scavenger, we can use that to infer the amount of oxidation of our analyte. let me just give you a quick view of how this works, okay? So we have our protein in solution. We have adenine as one of our scavengers. Of course, we have oxygen in solution because just from the air. And we use FPOP to generate a burst of hydroxyl radicals. These hydroxyl radicals diffuse through solution. Some of them encounter the protein and oxidize the protein. Some of them encounter adenine and oxidize the adenine. And in the end, we have a concentration of our oxidized adenine. And we can measure the concentration of oxidized adenine based on the change in UV absorbance at 265 nanometers. So we see an 8 milliabsorbance unit change here at 265 nanometers based on the amount of oxidation we have of adenine. Now let's say we take that same system with the same amount of adenine, but now we add in another scavenger. In this case, this is MES, just a typical organic buffer that's used in a lot of proteins. So let's say we're wanting to see, does this monoclonal antibody have the same conformation in two different formulations? And one formulation has this scavenger in it. I make the same burst of hydroxyl radicals. The hydroxyl radicals diffuse through solution. Some of them go to the protein, some of them go to the adenine, but some of them go to this new MES buffer and react with that. And so now I have less oxidation of my protein, but I also have concomitantly less oxidation of my adenine scavenger. And I can measure that photometrically by seeing a reduced change in my absorbance at 265 nanometers. Okay, so this tells me in my experiment that, hey, my protein saw less radical. So I can't compare this protein structure to my previous protein structure because the effective radical dose was not the same. But not only does this let me see that I have a problem in my experiment, this lets me fix the problem. So instead, I have my solution, I have my adenine, I still have my MES buffer, but I make more radical. I increase my UV fluence, so now I'm making more radical. And so now I get more oxidation of my protein, I get more oxidation of my MES, but I also get more oxidation of my adenine. I can easily measure the amount of oxidation of my adenine and see that it's now back to the same 8 milliabsorbance units that I had before in my first sample without the MES buffer. And so I can infer, based on the fact that the adenine absorbance or the adenine oxidation is the same, I can infer that my protein oxidation is the same. Okay, once I've done this compensation. So that's the theory behind it. Does it actually work? And the answer is yes. If it didn't, I wouldn't be telling you about it. So these are some data that we have. In blue is a protein and sodium phosphate buffer, which is a very poor radical scavenger, taken under particular FPOP conditions, and I get a UV absorbance change in adenine of 19 milliabsorbance units. If I change the buffer from sodium phosphate to MES buffer, okay, my change in absorbance of my adenine drops from 19 milliabsorbance units to 7. I keep the FPOP conditions the same, and you can see if you look at the amount of oxidation of each peptide, it drops. You have less oxidation of each peptide. The protein structure is the same, but I'm getting less oxidation because I've added in this MES scavenger. But if instead I take the same buffer and I increase the amount of oxidation by increasing the fluence of my light source to where the change in adenine absorbance that I get is effectively the same as what I got in the sodium phosphate buffer, now this compensated footprint that I've taken in MES buffer is statistically indistinguishable from the one I took in sodium phosphate buffer. And if you look at the oxidation products in sodium phosphate buffer versus MES buffer, they're essentially the same as well. And so you can use these radical dosimeters not only to see if you've exposed your protein to equivalent radical doses, but to compensate for changes in buffer scavenging. And that's where this UV dosimeter accessory that I showed you in my initial setup, that's where it comes into play, is this lets you actually make those measurements in real time. While yes, you can run your experiment and then just take the quenched solution and put it on nanodrop and measure the UV absorbance, and that works. If you find that you have different UV absorbance, that means you have to completely rerun your experiment. Using an inline radical dosimeter like we have here, we can measure it in real time. So this is a real experiment that we did. We measure our UV absorbance. We see a UV absorbance of our adenine at 79 MAU. We turn our laser on, and we see that it drops down here to about 70 MAU. But the problem is that we were wanting to compare this sample with one that we had made previously, where the difference in absorbance went from 79 to 66 MAU. So what we do is we turn up our laser fluence, and we keep turning up our laser fluence until we hit our adenine normalization target of 79 to 66 MAU. And once we've done that, now we know that we have sample that we can compare with our previous experiment. We've successfully compensated for differences in the formulation. So the adenine that we use, this adenine is used not in addition to the glutamine scavenger that I talked about, but it's used in place of some of it. So instead of 20 millimolar glutamine, we would use 17 millimolar glutamine and 1 millimolar adenine, because the adenine takes the place of some of the glutamine. One thing you have to be careful of, however, if you're using UV absorbance as a dosimeter, is you have to be careful of additives that can alter the UV absorbance upon oxidation. So the most common one that we run into is tris, which will show an increase in UV absorbance when it's oxidized, to the point to where we've actually run experiments using tris as our dosimeter instead of adenine, and they work very nicely. But it means that if you're going to use UV absorbance, while it's super convenient, very powerful, you always need to run buffer controls on new systems to make sure you test for UV absorbance changes before you rely on that data. Okay? But as long as you do that, inline UV dosimetry lets you do this in real time, and it works incredibly well. All right, and the last thing I want to talk about today is quenching. All right, after you've done the FPOP, you're going to deposit your sample into a quenching buffer that quenches the reaction. But you might ask, if you've been paying attention, which hopefully you have, you might ask, why do I need to quench? You told me the hydroxyl radicals last on the order of nanoseconds to microseconds. Why aren't they all gone? Why do I need to quench? And it's true, the hydroxyl radicals are all gone. You're not quenching hydroxyl radicals. Whenever you make hydroxyl radicals in oxygenated aqueous solution, you're going to make a stew of what we call secondary oxidants. These are other reactive oxygen species that are made from the initial hydroxyl radical that can still oxidize your protein, albeit oxidize it much more slowly, usually on the order of minutes to hours. And these secondary oxidants include superoxide or hydroxyperoxyl or hydroperoxyl radicals. They can include singlet oxygen. They can include the hydrogen peroxide, which is in the highest concentration because you added in hydrogen peroxide and you made hydrogen peroxide. So you have lots of hydrogen peroxide you need to get rid of. But then you also have what are called mixed organic hydroperoxides. These can be made from the reaction of hydroxyl radicals with just about any organic, including your analyte. And these mixed organic hydroperoxides can also oxidize your protein. And so you need to get rid of all of these. All of these can oxidize your protein, but again, they react much more slowly than hydroxyl radicals. So as long as you dump your solution into a quench immediately after FPOP, these secondary oxidants will all go away. This quench solution, however, needs to eliminate all of the secondary oxidants. So most commonly, quench solutions have two components. The first component is catalase. So we usually contain 1 micromolar of catalase tetramer or something very similar. And catalase is an enzyme that enzymatically converts hydrogen peroxide into water and stable triplet oxygen. So two things you don't need to worry about. And then the other additive is 70 millimolar methionine amide. So if catalase takes care of hydrogen peroxide, the methionine amide takes care of everything else. It acts as a competitive scavenger to react very efficiently with all of these secondary oxidants, usually via interaction through the thioether sulfur. And the reason we use methionine amide rather than methionine is that the methionine amide is both more water soluble and more stable than straight methionine. And so we typically use about 70 millimolar methionine amide. One thing that is important and that you always need to keep in mind as you're running FPOP, it is essential to always, always, always run unilluminated controls. Unilluminated controls are where you perform FPOP, you just never turn on the laser. And the reason why it's important to run unilluminated controls is the unilluminated controls not only tell you whether or not your quench worked, but it also tells you, did I let my protein sit too long in hydrogen peroxide before I started? Do I have oxidation of my protein that occurred during expression, purification, storage, etc.? Okay, it tells you the total of all of the background oxidation that you made throughout the entire lifetime of your protein experiment, so that not only can you tell whether or not I have too much, but you can also subtract that background oxidation from the oxidation that you get from FPOP. Because remember, the FPOP oxidation is telling you something about protein topography, protein surfaces. The background oxidation is not. And so you need to make sure you're always subtracting that from your results. So I want to tell you before I move on, until very recently, if you wanted to do the FPOP experiment, you had to build a bench like what I showed you earlier. So you had to have the laser and the optics and all of that stuff. Just this year, GenX has come out with a Fox oxidation platform, which pretty much does everything that I've just shown you in a benchtop format. The only real difference that matters is it's replaced the laser with an ultra-fast, high-pressure flash lamp. So that you don't have to have that laser, that excimer laser, and it's all much more compact. And it's all integrated. So it does everything that we talked about, flow system, photolysis, inline radical dosimetry. It even offers automated feedback control so that you can use the dosimetry to control the energy that you put on your flash lamp, so control your fluence of your light. And then an automated fraction collector to make sure that you can use the dosimetry to tell yourself when you actually want to start collecting sample. So if you're interested in FPOP but you don't want to build your own laser-based bench, this is an option that might be worth looking into. And then finally, I want to talk about post-oxidation sample handling. Okay, you've done the FPOP, you've quenched the solution. Now what? All right. So like most covalent labels, hydroxyl radical protein footprinting oxidation products are stable. However, the oxidized protein may not be. So for people that have worked in protein oxidation before, in things like monoclonal antibodies, you might know heavily oxidized proteins tend to be much more prone to aggregation and precipitation than unoxidized proteins. And the same is true of products that are generated from FPOP. So I usually recommend to people that are doing these experiments, immediately after quenching, go ahead and do your denaturation and digestion, because once you turn these into peptides, they're very stable. You can store these things in the freezer for months and the information will still be there. But if you let the intact oxidized protein sit, then in a lot of cases you'll start to see precipitation and you'll start to lose sample. And the important thing to keep in mind here is that you're not losing sample in a random fashion. You're preferentially losing all of your most heavily labeled protein, which means that you're losing information and you're skewing information away from the more heavily labeled samples. So you really want to make sure that you are digesting your protein as soon as possible afterwards. And if you have a sample that's really prone to aggregation, you may need to use some kind of chaotropes when you digest, something like guanidinium chloride or something like that, in order to keep this thing stable in solution while you do the digestion. For most systems that I've run in my lab, as long as I do the digestion immediately after quenching, I found that I can just do a thorough heat denaturation and digestion immediately after, and I don't have a problem with precipitation unless I let the protein sit. Once you've done your denaturation, it's important to keep in mind oxidation can change the rate of digestion in some areas, but it doesn't seem to change the digestion pattern when you're dealing with specific proteases. And so we've measured this to be true for trypsin and glu-C, and our preliminary data suggests that it's probably true for chymotrypsin. However, keep in mind, the oxidation can change the rate of digestion. So if you have a sample handling protocol where you like to try to do these ultra-fast 30-minute digestions that you're used to doing just for doing protein IDs, I implore you, don't try to do that with FPOP. You want to make sure you push these digestions to completion, as much to completion as possible, so that you're comparing the same digestion patterns between both of your samples. Because if you have differences in digestion patterns, significant differences in digestion patterns, it becomes very difficult to accurately measure differences in oxidation. So try not to use partial digestion. Try to get your digestion pushed as much to completion as possible. And then once you have this thing completely digested, you can do whatever sample processing you need. So if you need to do deglycosylation or any other sample processing, in general, you can definitely do it after proteolytic digestion. Just always keep in mind, you want to be sure that your sample workup does not have differential recovery between your oxidized and unoxidized samples. Okay, you want to make sure your recovery is the same so you don't skew your results. And then once you have your oxidized peptides, the rest is fairly simple. You do pretty standard LCMS and LCMS-MS using reverse phase C18 columns, and you'll end up with information that looks like this. Most commonly, I'll tell you, you do want to use a high-resolution mass spectrometer because the best way for you to identify oxidation products is through mass and through retention time. And so this is an example of one particular peptide and its oxidation products. You can see the peptide by itself. We've done very nice chromatography. We've got a nice, really sharp peak here at 11.87 minutes. But then if we look at the same mass plus the mass of one atom of oxygen, you can see we don't have one peak anymore. We have a lot of peaks here. What's going on? Well, remember, this one oxygen atom can be present at many different locations along this peptide. And it is. Each of these peaks represents a different oxidation product, okay? And so we're actually measuring oxidation at many different sites, all within this particular partially resolved envelope of peaks. And then if we look at the same peptide plus the addition of 2 oxygen atoms, you can see that while the diversity in the number of peaks has diminished somewhat, you still see multiple peaks, so multiple structural isomers of different oxidation products. And so the way that people normally report this information, you'll quantify based on the intensity of all of these different regions. But the way people report depends largely on what oxidation products you're seeing. So if you're mostly seeing your peptide by itself or your peptide plus 1 oxygen atom, so a single oxidation event, then monitoring the percent oxidation makes a lot of sense. Okay, and so for that, you would just take the total of the intensity of your oxidized peptide divided by the sum of the oxidized plus the unoxidized times 100%. That'll give you your percent oxidation. However, if you're seeing a significant amount of your peptide that has experienced multiple oxidation events in the same peptide, then it starts to make sense to use a metric that's a little more complicated than percent oxidation. And that metric is the average oxidation events per peptide. It works very similar to percent oxidation, except that the signal intensity for each of these oxidation products is weighted by the number of events it took to make the product. So for example, the plus 1 oxygen is multiplied by 1, but the plus 2 oxygen, this signal intensity is multiplied by 2 because it took 2 different events to make this metric. A plus 3 oxygen would be multiplied by 3 and so on and so forth, divided by the sum of all of the intensities of that peptide in both oxidized and unoxidized forms. Okay? And the reason why this is helpful is because as you get more intensity in the more heavily oxidized versions, percent oxidation starts to underreport the amount of oxidation that you have on a given peptide. All right, a few common themes in LCM in FPOP as we start to finish up here. And again, this is really the kind of thing where experience is the best teacher. So as you start to analyze these samples, you'll start to get more and more comfortable with what you're seeing. But just a few tips to help you get started. Oxidation products tend to elute very close to the unoxidized version of the peptide. So as you can see here, you'll notice in our chromatograms, almost all of the oxidation products are eluting very, very close to where the unoxidized peptide does. And that's typical. You see that most of the time. Most of them will elute slightly before the unoxidized peptide. You'll get a very small number that will elute after the unoxidized peptide, but they all elute very close to it. Oxidation elution profiles that are identical to the unoxidized peptide are a surefire indication that you have in-source oxidation happening in your electrospray source. So if your oxidized elution profile and your unoxidized elution profile overlap completely, then you need to lower your source voltage or move your emitter farther from the counter electrode or increase your nitrogen gas flow. Or one problem that a colleague ran across one time is they accidentally tried to do direct infusion without getting rid of all the hydrogen peroxide, and the hydrogen peroxide in the electrospray plume just oxidizes things like crazy. So things to keep in mind: if you see that elution time of your oxidized product is identical to the unoxidized peptide, check for in-source oxidation, because you almost certainly have it. One more thing to think about when you're trying to do MSMS of these things, because people always want to get down to higher and higher spatial resolutions. I don't just want to know how much oxidation I have on my peptide. I want to know how much oxidation I have on my amino acid. Keep in mind that standard ergodic MSMS methods like CID and HCD can usually tell you where an oxidation occurred if you're able to actually separate all of your isomers chromatographically to where you're only fragmenting one isomer. It can usually tell you where that oxidation occurred, but it can't tell you directly how much oxidation occurred there if you have a mixture. And that's because whenever you oxidize a peptide, that oxidation, that site of oxidation, is going to change the fragmentation pathway when you fragment by CID or HCD. So if you get good separation, LC separation of your oxidation products, you can sometimes quantify pretty well by peak area. But if you don't get good separation, or if you really need really precise quantification at the residue level, CID and HCD are not going to get you there. Fragmentation by ETD or ECD can be used to quantify unseparated mixtures of oxidation products, but that's a very complex topic. And UVPD may or may not work similarly to ETD. That's still not clear. And then finally, the longer your peptide, the harder it is to correctly identify and quantify sites of oxidation. So try to make sure that you choose a protease that's going to digest your protein into the sizes of peptides that are going to be most useful. A further discussion of hydroxyl radical protein footprinting data analysis interpretation. It's a very complex discussion. That will be the topic of our next meeting, December 9th. So I encourage you, if this is a topic that really interests you, please sign up. I will tell you off the bat, most commercially available software that people use for proteomics, it's good for identifying unoxidized peptides. It can be good for telling you that you have an oxidized peptide, but it's usually not going to be very good at telling you exactly where the oxidation occurred, and it's going to be very bad at trying to quantify exactly how much oxidation occurred at each site. So if you try to use that, please make sure you do a lot of manual auditing of the data, because there's going to be a lot of mistakes with most commercially available software. And with that, thank you all for your attention. If you have any questions, feel free to ask them now, or you feel free to contact me by email at jsharp@olemiss.edu.
I'll kick things off here. Can you just briefly explain the information content differences between FPOP and hydrogen deuterium exchange mass spec analysis?
Sure. So, the hydrogen deuterium exchange and FPOP are really measuring two very different things. The fact that they both measure protein structure to some extent often gets people to conflate the two techniques and think that they're overlapping or competing, but they're really measuring very different things. FPOP and other hydroxyl radical protein footprinting technologies are measuring amino acid side chain average solvent accessible surface area. Okay, and so it's really looking at protein topography, the surface of the protein and how that changes. Hydrogen deuterium exchange at its heart is really measuring the acidity and basicity of amide protons. Now, those are most strongly affected by hydrogen bonding. So things like secondary structure or other stable hydrogen bonds. They can be slightly affected by solvent accessibility, but it's really mostly hydrogen bonding networks that you're looking at with HD exchange. And so I would argue the two techniques are not competing with one another at all. They tell you two very different and complementary pieces of protein structural information. So HRPF is really looking at protein surfaces, protein topography. HD exchange is really looking at more like secondary structure and hydrogen bonding networks.